So here's part one from Unit B1, which is the bit all about cells. I'm just going to go through this fairly quickly. I haven't put the microscope bit in here, it's on a separate video, so I think the microscope bit is a little bit more complicated. So you might want to watch the microscope one, you might want to go straight to this. Okay, so hopefully you understand the basic sort of idea of cells, these building blocks, but I'm going to concentrate here on things that I think people commonly get wrong. So some of the organelles, which really just means little organs, I suppose, the bits that make up cells, bits that people tend to get wrong. So the nucleus, we know what it is, but sometimes people put these things like it controls the cell, it's the brain of the cell. I would put it contains genetic material, or it contains chromosomes, or it contains DNA. That's a better answer. The cell membrane will control what goes in and out of the cell. It actually does it on the basis of either size or even charge. If you know what ions are, charged particles, that's how it does it. So it controls things getting in and out. Inside of here you would have, very often they're just shown as little dots. They're called ribosomes, and what these do is make proteins. People forget about these a lot. They make proteins, they're the little dotty things. The things that kind of look a bit like a baguette or a French stick, those are our mitochondria. That's a plural actually. Mitochondrion is the singular, mitochondria is the plural. For that, remember that it is the site of aerobic respiration. It must be aerobic, okay? I know there is anaerobic respiration, that takes place somewhere else, I'll talk about that in a second. Aerobic respiration for the mitochondria. And the other bit that causes confusion for people is the cytoplasm. Colour. Really, when they discovered cells, what they would really do is they'd put things under a microscope and you couldn't see them a lot of the time because these things are basically water. They don't really show up as anything, they're quite clear. So you had to use a stain, which would be a chemical. So iodine is often used, you'll have used that lots of times. And iodine will stain parts of plant cells brown, for example. So that's a common stain to use. Another one is methylene blue. If you remember doing cheek cells, what methylene blue does is it stains the nuclei blue, okay? And a stain is used to increase contrast. Contrast means being able to see, use different colours. You know, if everything looked the same colour, or colourless, like it usually does, you can't really see a whole lot. If parts of the cell start to be different colours, it stands out. Okay. Now, when they first did these things, they saw the nucleus, that stain, but everything else looked like the same. They didn't have any, it didn't look like there was anything in there. And so they just used this word cytoplasm, which just meant everything that isn't in the nucleus. Now, we now know there's actually loads of stuff in there, but generally anything that isn't in the nucleus gets referred to as cytoplasm. And it's a bit of a difficult one to say what happens in there. So what I would say is the cytoplasm, chemical reactions happen here, and the easy one to give us an example is anaerobic respiration. That's probably the best example one to use, okay? Now, plant cells are pretty similar, they're larger. Let's use green. You can usually identify plant cells because they will have a regular shape to them, which is the cell wall. Now, again, here's a big problem. People forget about this. Basically, all cells, apart from animal cells, have a cell wall. Okay, so plant cells, bacteria cells, fungi cells, they all have cell walls. It's only animal cells that don't. The job of the cell wall is to prevent bursting. Okay, you can also, if you want, you could call it mechanical strength, but do not say that it protects the cell. It prevents it from bursting. Basically, water is going to go into these cells by osmosis and they're going to swell up, but you don't want it to burst. So the cell wall prevents the cell from bursting. The cell membrane will still be present, but it'll be pushed right up against the cell wall on the inside. There's still a nucleus. In the middle you have the vacuole, or the permanent vacuole, so it's there all the time, and this would contain cell sap, and this is kind of a sugary, salty liquid. It means the plant can take in water. Water will move in towards, if you remember osmosis, to wherever it's the saltiest or the most sugary, and it moves in there. They also have mitochondria, they also have ribosomes, exactly the same. But the key one that you would find that's different as well is the chloroplasts. Okay, chloroplasts absorb light for photosynthesis. Do not say that they trap light or catch light, they absorb light for photosynthesis. Okay. Fungi cells would basically have mostly the same kind of things that you'd find in animal cells and plant cells. They'd have a cell wall, for example, but you wouldn't really expect them to have chloroplasts because they're not plants. Okay, they get their food by another method. So fungal cells might look similar to plant cells in some way, but you wouldn't expect the chloroplasts in there. Different plant cells would, cells in the roots, for example, wouldn't have chloroplasts because they're underground. There's no light down there, so there's no point in them having chloroplasts, which is why the roots aren't usually green. But you could get sort of thrown those kind of questions. Now the bacterial cells, or to give them their proper name I suppose, called prokaryotes, are generally a lot smaller. They're about maybe in the sort of size of 1 micrometer. Animal cells are maybe about 20 micrometers, 20 to 50 maybe. Plant cells are maybe 50 to 70 micrometers. These things are much smaller. The name really translates into not having a nucleus. That's kind of what prokaryotes mean. So they'd be much smaller. I'm sort of exaggerating the size here. They still have a cell wall, which does the same kind of job as it does in plants. It stops it bursting. They still have ribosomes. They don't have a nucleus. What they have instead is one kind of big long loop of genetic material, DNA, a sort of big DNA chromosome if you like. They have smaller loops of DNA called plasmids. Okay, that's a little loop of DNA, and that's got a few genes on there which help the bacteria do various things. They still have a cell membrane. They don't have any mitochondria. Some of them might have one or two or several flagella, which are plural, basically a little tail that allows the bacteria to move around. But the main differences are at the nucleus. In fact, they're so different that all of these other cells, plants and animals and fungi, they have their own name too. They're called eukaryotes or eukaryotic cells. Okay. Now one last little bit on this, which is specialization of cells. This is a common mistake for people to make. Cells at first, I suppose you could call them stem cells, why not, because it comes up in chapter B2. A stem cell is a basic kind of cell. It's not done anything yet, it hasn't changed into anything. When it changes, we say the cell has become specialized. So it might become a blood cell, it might become a bone cell, it might become a nerve cell, or whatever it may become. The process where it becomes specialized is called differentiation. So if you like differentiation, that's the process. Okay, and sometimes people mix these two things up, so that cells undergo differentiation and become specialized. So in the second summary video we'll look at some of the bits on active transport, osmosis and diffusion, which probably are the bits that are quite difficult.